Okay, so when Min asked me to give this talk, I didn't realize how intimidating and overwhelming of a talk it was going to be to put together. So I'm gonna change the title slightly because also I realized that yesterday we took a lot of you from never doing scattering before to learning neutron spin echo in about 4 hours, which is throwing you into the deep end of understanding things. I've been now doing spin echo for about 9 years, and I'm still learning things as I go, so we understand this is overwhelming. So I'm gonna give you a little bit of some things that I've learned along the way, and how did I get here, and why do I still like doing dynamic studies, and hopefully entertain you, because I realize it's 9 o'clock in the morning and you all look like you are zombies. All right, okay, so how on earth did I get here, right? We get a lot of questions about where do people come from. I am not a physicist by training, I am a chemical engineer. I have done research in everything from proteins to polymers to membranes, so all of the things that we're talking about today, I have done some research in them. So for those of you who are protein people, I started out doing protein stability assays and figuring out how we can measure that proteins are actually stable. This is an SDS page gel, so if anyone here, I know we all know what proteins are, but if you've never tried to purify one, this is kind of what it looks like when you're figuring it out. I realized it's really hard when the thing that's making your sample can die. So I decided that I didn't actually want to do biology research when I went to graduate school, because when your samples can die, it gets a lot more complicated. So I became a synthetic chemist. Turns out reactions can die also if you don't set them up correctly. And actually, I tell everyone, I got into neutron scattering. I started out as a synthetic polymer chemist. I got asked to make deuterated polymers for one of my colleagues. And I was like, why on earth do you want deuterated material? If you just wanted, regular old polymer, I can make you a kilogram. You're gonna be lucky if I can make you 500 milligrams when all is said and done. And what's sitting in these reactors here is actually some deuterated polymers I was synthesizing for someone who was coming here to NIST to do neutron scattering experiments. So after lots of blood, sweat, tears, and a lot of dry ice to try to keep this thing cold so it didn't run away and die, I was like, well, now that I've made these polymers, I want to see what on earth they're gonna do with them. And that's how I came to do my first neutron scattering experiment here at NIST. So then I came here as an NRC postdoc. It's a fellowship. I really like working here. And as I was finishing up my postdoc, the VSANS instrument was coming online, and I was offered a position to stay on as an instrument scientist. So here I am today. So I'm a SANS instrument scientist. I spend a lot of time thinking about structure, so why on earth am I here telling you about dynamics, and how did I go from purifying proteins to telling you about neutron spin echo, right? And the answer is somewhere around my 4th year of grad school, I had this existential crisis. You can see that I am by no means the artistic one in our family, but I was trying to understand structural evolutions in polymer micelles. They were doing all of these really weird things, and I had no idea why things were happening. It was defying all of my basic understanding of energetics, and I was like, this is gonna take more kinetic energy than there is in the sun. How on earth is this happening? What is wrong with my cartoons? All of my data tells me this is happening, and I can't figure out how on earth it's happening. So I started to have this existential crisis about my research and what on earth was going on. And I realized that, maybe these cartoons were a little bit too simple. Right? This is a very rough schematic diagram of a polymer micelle, but if we start thinking about polymers, and as we've heard about yesterday, and as you're learning about in your sessions, these are actually much more complicated structures, and these chains are always in motion. Right? So these aren't solid objects. They have all sorts of dynamic processes that they can undergo, and because they're always moving, they can do things that we don't necessarily think just from our simple cartoons. So I became really interested in thinking about dynamics, but I was a grad student, overwhelmed and not really sure where to go and what to think about. So looking for motivation as someone who was in polymers, I decided when you think about dynamics and polymers, the first name that comes to mind is de Gennes, right, for all of my polymer people in the audience. And looking for some inspiration, I decided that I was going to go and read his Nobel Prize address. Right? So it's easy to get really discouraged in science, and sometimes you go and look to the big guns for some motivation. So I was reading his Nobel Prize address, and he first starts off talking about how calling it condensed matter science makes it sound really intimidating. And maybe it's going to scare away students, which is true. As a postdoc, when I was signing my offer to join the condensed matter science group, I wasn't entirely sure I knew what I was signing up for, because calling it condensed matter science is a very physics-oriented view of what we do here the NCNR, and I know a lot of you aren't actually physicists, right? So how many of you here knew what condensed matter science was before you got here? Maybe a couple, right? But the rest of us kind of think about it as soft matter, yeah, or material science. Yeah, okay, so Dejen talks about this, and I was like, oh, okay, that makes a little bit more sense. But then the other thing he talks about in his Nobel Prize address is he goes through and he gives examples of what he considers. Oh, okay. Oh no. This is a surefire way that I'm either going to smack this or fade in and out of. Can you still hear me? Is this okay?
Yes, just for the recording purpose, but like he can turn off the amplifier. No, I think it's on the mic.
Okay. Cool. Good? Good. Okay. So in the rest of his Nobel Prize address, Dejen goes on to talk about what he considers good examples of soft matter, things that we're all interested in here. He talks about some of the phenomenon he was studying, and one of the examples he gives is this idea that we talked about yesterday in our membrane session, where if you observe a cell under phase contrast microscopy, so if you were to pull a red blood cell out of your body and put it on an optical microscope and watch it, you would see that these cells flicker. This was first observed in 1890. Before we even knew all of the components that made up cells, we knew that these structures were dynamic. And it's like, oh, well, that's pretty cool. And being a studious grad student, I immediately Googled it. And it turns out that courtesy of the internet and YouTube, we can watch this now too. Right? So as far back as 1890, it was known that whatever it is that makes up our biology in our bodies is actually a dynamic, moving, changing structure. And I became really fascinated with this idea of, how do we measure dynamics? And how do we relate it back to fundamental material properties? Why you're all here now, and why 9 years later, I'm still here at the NCNR, trying to learn about Spineko and what it can do to characterize materials. And part of the reason this blew my mind, right, is if you open a biology textbook, you see something that looks like this, right? So the first thing they teach you in biology about biomembranes is that the primary function of the membrane is to form a stable boundary. Period. End of story. But this boundary has to move, right? And if we think about all of these cell functions and all of the things that is happening in our cells as we sit here and talk, in all of these cartoons, we're still looking at the cell membrane. But now it has to be able to dynamically rearrange, reshape, and reform, right? So now if I look at you as people who are interested in materials and I say, all right, so I need you to go make me material that's stiff enough to form a stable boundary, but not so stiff. It also has to be flexible because I need it to be able to adopt all of these crazy shapes and change structures dynamically when I want it to, but it can't fall apart. You would look at me like I was crazy. You'd be like, all right, forget this. I'm done with material science. I'm going to go do something else with my PhD. But yet somehow, nature has figured out ways to do this. So we spend a lot of time, Michi and I, who I've now been working with for a long time and who has taught me everything I know about Spineko, thinking about how do we then measure these dynamics and these material properties of membranes, and once we have these tools, how can we understand how lipid composition influences them? All right, so that's how I got here. And now I'm supposed to give you a case study about going from ideas to papers. And I don't know about you, but writing papers is not why I became a scientist. It's probably one of my least favorite things to do. When all else fails, PhD comics reminds us about what it actually means to be a scientist, and sometimes it's important to laugh. So I was trying to figure out what on earth am I going to tell you about for a case study today. And yesterday, Bala told us about some of his favorite examples of neutron spin echo experiments. So I decided to show you a case study of one of my favorite examples of science, but along the way show you that this is by no means a linear process. And at least for me, it takes a long time to go from having an idea to being able to write the paper. So the example I'm going to give you is thinking about this system here that I published in 2021. This is some beautiful cover art made for me by Ryan Murphy, and you can see on the front of Soft Matter, we have a nice cartoon of what looks like a fluctuating membrane. So it's always a good day when journals like your article enough to then give you the front cover of it, right? So that was a happy day. All right, so step one, what is the big picture motivation? You know, it's easy to get lost in the weeds of our scientific project, but at the end of the day, we have to be able to explain to everyone, particularly our bosses and the funding agencies, why we're doing what we're doing. So, broad idea of why are we trying to understand membrane dynamics, and in this case, we're looking at the effects of peptides on these membrane dynamics, is that actually biomembranes are really complex structures. Right? Even if we look at the simplest organelle, in this case it's a synaptic vesicle or a signaling molecule, this is reconstructed based on lots of NMR, cryo-TEM, and SAX data. This is the simplest possible biomembrane you can make in biology, and you can already see that's pretty gosh darn complicated. Right? So this yellow part here is the lipid membrane. That's everything that we've been talking about, but it's also full of all of this other stuff. And you know, I can make some rough cartoon approximations of this, but you know, how do we think about now understanding the dynamics of something that looks like this, right? Because if I now think about a dynamic tug of war between the soft lipid matrix and the proteins are always going to win. A protein misfolding is pretty gosh darn bad for biological function. So really, what this membrane is a mixture of really hard and really soft things. So how does this heterogeneity, right? I've got not just a soft thing, I've got soft things mixed with hard things, and the hard things of varying stiffness. How does this heterogeneity affect the membrane dynamics that we're interested in? Big picture idea. Great. Well, there's about 30,000 different lipids and about 30,000 different proteins, so that quickly becomes a really complicated problem that we're not going to solve in one experiment. If you can, please come talk to me. I haven't gotten there yet, right? So now what we need to do is sit down and write a proposal. So I like to think of it as writing a SMART proposal goal, right? So if those of you who are in sports or you know any sort of motivational thing, they'll tell you about writing SMART goals, where SMART is an acronym that stands for specific, measurable, achievable, relevant, time-bound. That means some things to, in contexts of goals in general, but we're going to talk about it a little bit in the terms of writing proposals. Also because you're going to hear more about today at Working Lunch, specifically writing a proposal. Right, so we have a big picture idea of why this is important, right? We want to understand dynamic heterogeneity, but I'm not going to solve that in one experiment. I want to write a specific experiment to address that bigger problem, preferably something that I can actually measure with the technique I'm trying to do. And I've done the calculations in the back of the envelope estimates to make sure that spin echo is actually the right technique, because as we learned, spin echo is the last thing you do. If you come and tell me that your time scale is going to take milliseconds, I'm going to say, that's really cool, but it's not something I can achieve on a spin echo instrument. Or if you say this is only going to take picoseconds, again, that's cool, but maybe spin echo isn't the right technique. So figuring out if it's achievable and relevant for the timescales that you want to do. And then when you think about time-bound, also think about that this is a finite resource, right? So if you say, I have this really great idea, it's going to take 365 days of Spinnaker time. Cool. We don't have 365 days of Spinnaker time. So thinking about all of this, we want to make sure that it's also a reasonable request in time, because we want to give as many of you time as possible. Right? So these are kind of some of the things you want to start to think about when you sit down and write a proposal. It's making sure that it's measurable, I can do it, I've done all of my homework so I know what it is going to be, it's going to be achievable in a reasonable amount of time. It's I have this broad goal of understanding heterogeneity. So for this example, the first proposal we wrote was this idea of looking at how having a very simple peptide affects the out-of-plane fluctuations of the membrane. Okay, so now this is now a much more specific goal that's motivated by some theoretical calculations that were done by Helfrich and Jacobson in 1990, where they proposed that if you could have a membrane do these out-of-plane thickness fluctuations, then that would be an easier way, or it would help facilitate protein-protein interactions, because it can bring them in closer contact together, and it would allow this peptide to form a dimer. All right, so now here's my specific thing that I want to look at. I have a specific peptide I want to look at. I have a measurable thing. I want to measure thickness fluctuations. I'm going to pick a model system, and I'm going to write a proposal around this, right? Because this is an interesting case either way, right? We have this hypothesis, there's some theoretical predictions, but also in my naive, simple mind, if I have a membrane doing this and I put this really rigid thing in it, I can imagine at some point I'm going to just pin those two leaflets together. So either way, what we measure is interesting, which is an ideal experiment, right? Your results are interesting in either case, and you have some hypotheses, and you can start to think about it from both perspectives. So we set out to try to measure this specific goal. If we take a lipid membrane, we put in low concentrations of this peptide, will we see an enhancement in these fluctuations to facilitate dimer formation, or will we pin these leaflets together and completely suppress the dynamics? That is our hypothesis. That was the goal of the proposal. So I went back and I found when we first put this proposal in, and you will see it was done in 2016. That paper came out in 2021. So it took us a while. As part of putting together the grant to be able to build a new Spinnaker, we had to collect a lot of statistics, and Michi and Antonio worked really hard to figure out how long it took to go from proposals to publications, and it turns out I average about 5 or 6 years, which I realize as a PhD student, that's like a terrifying number to hear. But that's why we're here, because we figure out the hard stuff and then help you get there maybe a little bit faster than I do. But again, I'm also not all that great at writing papers, and Michi's sitting in the back corner laughing and nodding his head because he knows that's true. Okay, so I wrote my proposal. I had a clear goal. I explained it. I told the reviewers why I wanted to do this. The reviewers said, okay, yeah, this is cool. It's doable. It's a smart proposal goal. It's achievable. It's a reasonable amount of time. You're not requesting all of this Spinnaker time in the year for this facility. Let's go for it. All right, so now we've got beam time. Great. Do your homework ahead of time. Spinnaker is the last thing we do, right? And as we learned about with most scattering things, this is one of my favorite XKCD cartoons as a scatterer, right? We do a lot of Fourier transforms in scattering, but you learned about how we have the phase problem in neutron scattering, so you can never completely recover your initial input. So that's bad news for the cat, unless you know you started out with a cat. So you want to come here to do your experiments with as much information as you can possibly bring, because the more information you can give me at the beginning, the more likely we are to get your cat back out at the end. Okay? So you want to do your homework before you even get here. Spinnaker is going to be the last thing we do, right? So read the literature. For this one simple or specific project, right? Here is all of the different theories that could potentially explain what we might expect to see. That's just the theory. You also want to do your review of literature for the experiments. Has anyone done this before? Right, I actually pulled this. This is me making these slides this morning in my first cup of coffee, because my presentations are also made with blood, sweat, tears, and caffeine. Right, this is the stack of papers I pulled out of my filing cabinet for this proposal. These are all experimental references that I was reading to try to guide my experiment design to find the conditions that I wanted to be able to measure. So you're going to do all of your literature search, do your homework, have your goals, know what you want to do, and then characterize the structure first. Right? And in this case, I was looking at dimerization in a lipid membrane. So I have two components here. I need to be able to look at my peptide and my lipid, and I want to make sure that my peptide is doing what I think it is, my lipid is doing what it is, and this meets the goal of my original proposal. So here I wanted to make sure that I am actually forming this conducting dimeric form of the protein. It's forming the right structure and doing what I expect it to do. But I also want to make sure I haven't put in enough peptide that it's affecting the ensemble average structure of the lipid bilayer. Because I want to look at the peptide effects, not the effects of the peptide on the structure in terms of the dynamics. So we're trying to find these out-of-plane fluctuations and facilitating dimer formation. So I characterize all of this ahead of time. And then you go and you do your experiment. Because you've done all of your homework, you're confident that you know what you want to do, you have your samples well characterized. But the important thing about going to do any experiment at a large-scale national lab or facility is you don't just have plan 4A, what your original proposal was. You have backup plans B, C, D, E, F, G, H, etc. Right? Because as my boss always likes to say, no plan withstands first contact with the neutrons. So you might have the best proposal idea in the world, but maybe the FedEx shipping center at Charles de Gaulle Airport breaks down and your samples get lost. Maybe you accidentally contaminate your sample while you're using the rotovap at a lab and your sample turns orange. Maybe the Neutron instrument is so good, you're going to collect data a lot faster than you originally estimated, so you can run more experiments than you originally thought you could. Or maybe the experiment after yours gets canceled, and your friendly local contact says, hey, I have an extra day of beam time. Do you have anything else you want to run? So you don't just plan for A, you plan for lots of things. Because ideally we do A, but if not, this is a finite resource at any scattering facility. You want to make sure you use the time to the best of your ability. So when we go into experiments elsewhere, it looks like we're moving the labs. I ship like boxes and boxes of samples. Sometimes you lug several hundred pounds of equipment through airports. I'm not suggesting you do that, but just make sure that you have a backup plan, because either way, I guarantee you the experiment is not going to go the way that you planned it, and if it does, please come and talk to me, because I've never really had it work out the way I wanted to. So let me know what I'm doing wrong. Okay? And then make sure you do all of your complementary characterization samples on the exact same samples you ran NSE on. You will thank yourself later. Right, so if you need to do dynamic light scattering or small angle neutron scattering or SAX or DSC, anything and everything you need to understand what that sample is, make sure you do it on your NSE samples too. We work really hard at scattering facilities to get you all the complementary characterization information that you need to be able to characterize this. You want to know exactly what that sample was you put in the Spinnaco, because you might get to come once or twice a year to do that experiment. So when you want to know everything you possibly can about that sample. Let us know ahead of time. We'll help you get access to the complementary characterization equipment that you need to do it. All right, so you've got data. Ooh, we're running behind schedule. I will move a little bit faster. Now it's time to analyze the data. Always start as simple as possible. Please, please, please look at your data before you start trying to fit it. So this is some of the data that came out of the experiment. I haven't given you any labels, because if we just look at the data that comes out, this is the relaxation rate divided by the Q. These are for different samples. They look different. I haven't even done any analysis in terms of fitting the data. I'm just looking at it. Please look at your data before you start trying to fit it. And now, if I start to label the samples, This is my pure lipid membrane. This is increasing peptide concentrations. I see differences depending on how much peptide I put in the sample. Great. I haven't tried to analyze it yet in terms of quantitative analysis, but qualitatively I can already start to get some information. So we're going to start as simple as possible, and then we're going to build in complexity. Okay, I see differences when I add peptides. It seems to depend on peptide concentration. What are some existing things in the literature that I can do to try to analyze this data to start to pull out more quantitative information? These solid lines are now fits to this data based on what was published in terms of analyzing these thickness fluctuation signals, so these out-of-plane dynamic signals where I have some sort of information on the amplitude of the fluctuation based on the peak width in Q space. So what is the length scale associated with this? I have some information on the time scale or the relaxation time based on the peak height. But then also, always be critical of your own results and your own fits, because the worst thing that could ever happen to you as an experimentalist is if someone comes and repeats your measurements later on, right? So I have this pink curve here, but maybe it's this green curve. I have the tendency to be overly open and honest in explaining things in my papers, and in the paper on this, we said, yeah, well, we fit it with this based on these assumptions, but we can't rule out this other thing. Gets me into trouble all the time with reviewers. But if anyone ever comes and does this again and actually gets that it's the green peak, at least I know that I said, well, it could actually be the green peak also. So I've covered my back there, but you know, science is about learning and expanding, and it's okay not to have all of the answers, right? So as you're analyzing your data, maybe it makes some sense, maybe it doesn't. Another PhD comic that is helpful for remembering things. So maybe you're not 100% convinced, or maybe things didn't work exactly as you want to. So maybe you go back and you need to repeat this and get some more data, right? So this looks pretty good here at low peptide concentrations. I don't really know what's happening here. Was this sample bad? Does it make sense? I was expecting to see an enhancement in these fluctuations. Now it looks like I completely suppressed them. What on earth is going on? So I go and I do some more experiments, another set of controls to be able to figure out what's going on. And then you have to write the paper. I know I'm almost out of time, so I'm going to quickly go through this. This XKCD was shown to me actually by someone in the NIST Public Affairs Office, so if you haven't seen this, I highly recommend it. But the important thing is you know, they broadly classify scientific papers. The top left is we put a camera somewhere new, and for NSE that's usually true. Right? But I would like to say that it's not just a camera, it's a movie camera, because now we're not just figuring out what things look like, we're figuring out how they move. So we can zoom in and start to understand new things. That's why you're here, to learn how to be able to do these and understand these motions and write up a paper, right? So this is kind of the broad general things of at least how I try to approach an experiment, with the understanding that this also took me almost 6 years. Hopefully you will do it faster and more efficiently than I did. Hopefully this is at least somewhat entertaining for you and giving you some guidelines of things to think about when you go from having your big picture idea to then actually designing a Spinnaker experiment. And I'm happy to answer any questions that you have. Antonio.
Can you tell us how much samples you used for these experiments? And quantity,
The volume or?
Oh, okay. Yes. So it depends on the measurement you're doing. So at the end of the day, it ended up being, in the paper, there's about 12 samples. For any given experiment, I typically did 3 or 4. These are lipid vesicles in a solvent. So I needed, depending on the contrast, I needed on the order of 2 mLs to 6 mLs for thickness fluctuations. Those are harder to measure. You don't have as much signal. It takes longer. It takes a lot of deuterated lipid. We're some of Avanti Polar Lipid's favorite customers. So each of those samples was probably about $600 worth of lipid. So this is expensive in terms of materials, but then the other thing to keep in mind is that a day of beam time at NIST is the equivalent of about $5,000. So then a 7-day experiment is about $35,000. So by comparison,
$600 for lipids on a $5,000 measurement isn't actually so bad, which brings to an important point. If you need a deuterated solvent or something for your sample that you don't have and it's what you need to make your experiment possible, please tell us ahead of time. We have D2O here. If D2O is what's preventing you from designing your best experiment or a deuterated alcohol, please email us. If it's $50 of solvent, we might have it here and we will give it to you if it will make your experiments better. Does that make sense? Yamali Hernandez works very, very hard to try to make sure that we can help you get the data that you need.
Were there like 30 mg per mil, 50 mg per mil? Can you estimate or?
Probably on the order of 100 mg per mil. So with any experiment, you're balancing sample amount and count time. Right? The more sample I can put in the beam, the less count time I can have. So in that case, since we had the material, we used a thick path length. If you have to go to a thinner path length and have less sample, then it's going to take longer to count and you're going to run fewer samples. It's always an optimization. Unfortunately, the thing that we always have to decide. So that's why when you do your experiments, please talk to us ahead of time. This is part of why we always ask you to do the structure and run SANS first, because we can look at the SANS data and say, oh, you don't stand a chance of getting data without having 4 mLs of 100 mg per mil sample, or your sample scatters really strongly. You can probably get away with a milliliter or 2. Any other questions?
All right, all good?
Cool.
Thank you,
Yes.
thank you very much.